Bryan Jones. How are you this morning?
Good, thank you.
Brought
How
your
are you?
coffee.
Brought my coffee. Have to have the coffee.
Good to see you.
Thank you.
Hi. Have a seat. So I met Bryan recently through Chris Johnson, and it was a natural flow of conversation from the things that you saw already this morning. So you're having the same flow here on stage as we've experienced in real life, if that's a comparison. I think we had real life this morning. We had this kind of incredible, I think, conversation on the telephone or the phone. And
Yeah, that was fun.
the reason that Chris recommended me to Bryan is that Bryan is a world expert in the eye-brain story. You know, what is the eye-brain connection? It's called a retina. And how does it work? And what kind of processing happens there? And Bryan knows a lot about this. So welcome.
Thank you.
And this is a not accidental step toward trying to help our guys this afternoon understand more and more about the various ways of coming at the question of how to create a new computing paradigm, which we're calling pictorial processing.
Right.
So let's start with the simple things, because there are probably a lot of folks in this audience who don't have any idea what we're going to be doing up here.
Right.
Why the retina? What's interesting about the retina?
So the retina,
And describe it as an eye-brain thing for
yeah.
everyone.
So most of us have retinas. How many people in the room are visually impaired? Anybody? AMD, retinitis pigmentosa, cataracts. You know. So there's a couple of people. This pretty good. Eventually, sort of, if we get old enough, we'll all have visual deficits, and we get used to sort of navigating our world visually. But what does visually mean? Well, we have these sensors at the backs of our eyes that we take in information with, and these sensors, a lot of people like to describe them as cameras, but they're not really cameras. They're more flow processors, right? And they take in the information in our visual scene, and they do a lot of pre-processing for it, and then split those data streams up and send them to the appropriate areas in cortex and brain. I guess we can probably start going through some of this stuff. So some of the stuff I'm going to talk about here was pioneered by my scientific mentor, Robert Mark. And a lot of the data I'm going to show comes from his lab. He's retired this year and is now growing apples for apple cider. But what we're showing you here is sort of a fly-through of our data set. This is the world's first connectome that Chris Johnson alluded to earlier. Wow. This is a segmentation. It turns out there's no empty spaces in the retina. We're flying through the glia. So in between all these neuronal connections, there are glia cells. And it turns out the glia have substantial importance to helping keep the neurons alive and happy, and also probably processing some data. So this is looking at the underside of the retina right here. This is the ganglion cell side, and I'll show you what this means here in just a second. But that was just sort of a quick through to show you the level of complexity of this thing that we call a connectome. And our lab was one of the first on the planet to jump into the space. This, what you're seeing here, is the first connectome on the planet, and actually the only connectome on the planet with all of the resolution required to see the connections.
So define connectome for everybody.
So a connectome, sort of mathematically, you can sort of think of it as the edge node graph function of how information flows through the retina. Different people take connectomics to mean different things. It operates at different scales. Some people operate at medical imaging scales, and they're looking at diffusion tensor imaging, weighted images that look at white matter flows through brain. This data here, as I'll show you in a minute, was captured at much higher resolution, because we're actually interested in the literal connections, the synaptic and gap junction connections, and I'll get back to you to those in a minute. So we'll just walk through some anatomy. This is my wife's eye, and the sort of the blue sclera, in the pupil. This is what we're all sort of familiar with, but this is my eye, and so this is an infrared view of my retina at the back of my eye, And so when you look through the pupil and you look at the back of the eye, you're actually looking at brain tissue. So when we develop, our eyes sort of come off as stalks from the brain and come out and develop into eyeballs,
Yeah.
right, with the sensory network. But it is brain tissue that you're actually looking at the back of the eye there. So
That's a really critical point, I think. So the retina represents the meeting of the brain, and the brain is actually in development, sends out these orbs, these extensions, which are pretty far removed from the main mass of the brain. But it is a physical part of the brain which is in the retina,
Absolutely.
connecting with the outside world.
Absolutely. And then, as we're going to show you here, it becomes a highly specialized sensory device
Right.
that then feeds into the brain.
Right.
So the structure at the front of the eye is the cornea. That's what they operate on when you get LASIK. You can see the lens behind it. That's where you get cataracts. That's what we can do to replace it. But the pink structure at the back is this retina. It lines the back of the eye. And when that's broken in us, we can't fix it right now. So there's a lot of research that's going into that arena. There are other organisms with more complex retinas, actually, that can fix their own retinas. And that would be a lot of fun to study, but we're not funded to do that. At any rate, so
What's one animal that has that?
zebrafish, goldfish,
Zebrafish,
turtles,
huh?
salamanders.
Okay.
You know, lots of vertebrates that actually, so part of the reason that we study the mammalian retina is that it's a simpler retina, right? And so if you look at bird retinas or turtle retinas, they're far more sophisticated. So if we zoom in here, so this is a primate retina, and so what you're seeing is if you go from the left side of the image to the right, that's where light flows. So our retinas are actually transparent, and so photons go through the entire retinas to hit these sort of cone and rod photoreceptors at the back of the eye that you can see there. Is there a laser pointer? Or? It doesn't matter. So we see in red, green, and blue, unless we're colorblind. But turtles see in nominally 7 spectral channels, right? So they have more photoreceptors and more kinds of horizontal cells and bipolar cells. We're going to just turn this one sideways and then just show you sort of a quick summary. This is a nominal accounting of the types of cells that are in.
So the light will be coming in
From the bottom now.
That's what I thought, okay.
Yeah, so it's backwards from the way you'd think of it. If you were thinking you would design a sensory device, you'd put the actual light sensors at the front of the eye. And actually octopus eyes are designed like that. But there's some good reasons for turning the retina around metabolically. So the interface in between the photoreceptors and the retinal pigment epithelium is the area of highest metabolic activity in your body. And so you need to be able to supply the photoreceptors and take away waste products. And that's the vasculature at the very top of the eye there. At least that's the way, that's our rationale for how that works. So in a mammalian, you've got nominally 70 to probably 100 kinds of neurons in our brains. And something like a goldfish would have 220.
So
Right.
we're back for a minute to our prior conversation where
Yeah.
brain-inspired is a nice term, but in fact, if you look at chips today, they have one type.
Right.
So you're saying how many types? 100 for humans?
So
200 for-
So in a microprocessor, we use gates and transistors and
Right.
and ways of processing how information flows. In the nervous system, we use different kinds of signals. We use neurons and glia that do different kinds of gating functions, right? Some are electrical capacitors, others function as sort of analog processors, like the
Smooth signals.
photoreceptors, and bipolar cells. And then others, like the ganglion cells, function as sort of bursting, sort of
Spiking.
more binary
Yeah,
signals.
which is now more common in new chips, in non-human
correct.
chips.
Yeah.
And analog is coming back, I'll mention. And some of the people we've
Yeah.
talked to about pictorial processing say, well, are you talking about analog there? So that
Right.
it may be that that's involved somehow.
Yeah, so,
In this picture that we're looking at right now, if we were computer guys, guess we are, so we're seeing a pre-processing system here in the retina,
Yes,
which
sir.
actually is a computer of its own,
Yes, sir.
which does compute functions in a pictorial way prior to sending a limited amount of information to the brain.
Yeah, so when, if we go, can we go back with this? Yes, oop, went too far, sorry. So when images come in to the left of the eye and are projected back on the retina, that is an image.
Yep.
And so what then starts happening is the retina detects that and breaks the image down into, it starts derivatizing the image. It starts basically breaking it into a series of voltage changes that then start separating the signal.
So you heard your pals this morning talking about visualizing visualizations,
Right,
and abstracting the information from that.
right.
Is that happening here?
That's exactly happening here.
Wow.
So we have
Wow.
photoreceptors that start taking apart
That's
the
amazing.
color information.
That's what's happening here.
Absolutely.
Okay.
So color information is broken down at the very first synapse. And then you start breaking down form and direction and contrast and luminance.
Features.
And then there's other longer lasting signals like circadian information. And all of this sort of, as you get through the retina, so as we're looking top to the bottom here, the ganglion cells at the bottom are the output cells of the retina. And there are at least 20, probably 40 classes of cells in our eyes. And that's probably 40 separate streams of information that are streaming different derivatized types of data out of the retina and projecting it to different areas in cortex and subcortex.
So all this sorting, right?
Yeah,
And filtering
right,
and pre-processing
right.
and abstracting to prepare the information before it reaches the brain.
Right. So you need to process color and form and contrast and direction to start putting together the derivatives so that you can start assembling faces,
Yeah.
for instance, or start extracting other higher level meaning from that.
Have you ever done the math on, so when you think of a normal human being, walking around 24 hours.
Yeah.
Well, not 24, 12 hours. And the amount of information that comes in through the eyes.
Yeah.
Do you know any numbers about that?
So
I'm thinking about, like, in your drawing,
Yeah.
the inputs,
right.
roughly how many bytes are coming in?
Oh gosh, this is actually hard to estimate. So there are a bunch of people that are throwing around numbers based upon the number of photoreceptors, and they try and extrapolate an interpretation on the number of photoreceptors is the number of pixels, and so they say, oh, okay, you know.
But I mean, just coming, not what gets, not the next level of processing,
Right.
right? That level, but literally the camera level. So you would know, if you were a camera,
right.
how many
That's just what I'm saying.
photons came in.
So they say, you know,
You know,
they may say, all right, well, is the human retina an 80 megapixel sensor, you know? And then there's 80 megapixels over time, right? So there's this concept of flicker fusion frequency. So back in the early
Bryan, I
days
want to
of
go even
film.
further in front
Okay,
than that.
all right.
Because I think these are all filter steps, like
Yes.
how many receptors you've got as a
Absolutely.
filter step.
Yeah.
But the actual number of bits that are coming into your eye, right? In the evolution of the human species, some decisions were made, or whatever, however you want to put it. But,
Yes.
but it would be, maybe we can't do it right now, but it would be a really interesting number to know, total number of bits incoming for you, for your daily life, and then compared to total number of bits that get through all these filters and processes and end up coming down the optic
Out
nerve.
the optic nerve, yeah.
What's the reduction factor between those two?
That's a good question. I don't think we know.
That'd be a really interesting number.
It would.
Yeah.
And there are people that try and study the output of the retina. There's a guy at Stanford, E.J. Chichalinsky, and a number of other people that place these retinas on multi-electrode arrays with thousands of electrodes and present them stimulus and
Yeah.
try and record
Yeah,
from
right.
them and get estimates
Yeah.
like that. But this goes back, I mean, we've been studying the retina this is 1892. This is the work of a guy named Ramon y Cajal, famous anatomist. And so these are all retinal neurons from a vertebrate mammalian. And so what Ramon y Cajal did is he silver stained all of these and then drew them in this thing called a camera lucida. The important thing to realize here, though, is that all of these neurons that he drew are sandwiched together, right? So there's a couple of connection layers that our previous speaker noted in hippocampus. We study retina because it's sort of a simpler version. It's more compact. All the circuitry is present, and it gives us sort of circuit topologies that we can start understanding how information flows through. The hard part is we've been doing this for 150 years, and it's not easy. And so Ramon y Cajal started with the anatomy, the raw anatomy, and we're interested in the network analysis. So from a simple network analysis, in a basic retina, you have a cone photoreceptor, and that information is sort of in an analog signal sent to a bipolar cell, right? And the bipolar cell, in an analog fashion, stimulates amacrine cells and ganglion cells. And then amacrine cells feed back onto bipolar cells and ganglion cells and themselves, right?
So you have a feedback loop here.
There's a bunch of feedback loops,
Okay.
it turns out.
Yep.
And so retinas are built like really high-end amplifiers that tune spectral
Yeah,
responses, right? But this is a simplistic, this is the best case, simplest scenario
Right.
for how that works. The reality is a bit more complex, and we know that because we're taking this connectomics approach, and we want to know what the patterns are and the overall network topology. So there was this guy, Frank Harare, who unfortunately died back in 2005, and he basically did the math to figure out potential combinations of connections among things. So if you have 3 separate things, there are 8 different ways that you can connect these 3 separate things, right? It doesn't seem that way, but it is. And if you allow directed graphs, there are 64 different ways that you can connect these 3 separate things, A, B, and C. And when we did the math, I didn't believe it. So I plotted them out. And it's true, there are 64 different ways that you can plug these 3 things together. And if you allow reentrant loops, like what happens in amacrine cells, there are 512 different ways that you can plug these 3 things together. So if you start thinking about the brain and the retina, it becomes crazy. And you can start looking at some numbers. So the number of seconds since the beginning of time is 4.0 times 10 to the 17th. Okay? The number of atoms in the universe is 1.0 times 10 to the 80. The Shannon number, which is the total number of potential moves in a game of chess, right, is 1.0 times 10 to the 123rd. Part of this is driven by the number of separate objects or the number of classes of connectors that you have in there. And in a retina, if we say there's nominally 70, there are 9.0 times 10 to the 1,473 different potential network topologies that you can come up with in retina. And in brain, it's 9.0 times 10 to the 300,728, right? And that last number is why the singularity is never going to work mathematically. But that's another talk. I actually feel pretty, I get pretty hot about that.
I do too.
Yeah, so.
Not a big fan.
Yeah, maybe we can talk about that another time. So because the numbers are so big, the only way to understand the topology is to map it, is to go in and actually map it. So we use in-dimensional metabolic clustering approaches to label the cell classes. And I was pleased to hear Valerio's talk, because Valerio, we need to talk about some ways of clustering metabolism. That'll be fun. And then in between all these, we do big data reconstruction of ultrastructure. And so that means what we do is we basically go in and look at 2 nanometers per pixel. So Kristen Harris at the University of Texas showed that you needed this resolution to be able to see the synapses, to be able to see the vesicles and the gap junctions. And so that sort of defined the absolute minimum magnification that we had, which meant that our data sets were going to be really big. So each one of these slices is about 90 nanometers thick. So the wavelength of light, green light, is like 340 nanometers. So literally, if you took one of these slices and you turned it end on, it would disappear, right? That's how thin each one of these slices is. And then we labeled them with antibodies to look at their metabolism, and then we go through and we trace and reconstruct their connections. And we do it with a level of detail that basically allows us to pull out individual cells and start looking at all the potential connections on them. So this is a wide field cone bipolar cell, one of the 70 to 100 types. We have 194 ribbon synapses, so analog connections onto this bipolar cell. 274 postsynaptic densities, so there's 274 neurons that are talking to it. There are 20 gap junctions, which are like electrical connections between cells. 127 adherens, which are other cell type connections. And then 66 conventional and cistern synapses, which are again analog digital connections from amacrine cells. So this is the level of resolution that we need to be able to pull out the network topologies.
Wow. I'm glad you're doing this instead of me.
Oh, it's fun though. I mean, you know, as long as we can keep funding to do this. So that cone bipolar cell was a simple cell. This is a more complex cell. This is the most complex cell in our retina, the A2 amacrine cell. And this is its network diagram. If you look,
One cell.
one cell, it's kind of hard to.
This is one cell.
So this yellow cell at the center of the network right there is the A2 amacrine cell. And this is 2 synaptic hops away from the A2 amacrine cell.
So when we talk about brain-inspired or nerve-inspired chips,
yeah.
forget it.
Well, no. So this is just engineering, right?
Right.
And so part of the problem is figuring out what this wiring diagram means.
Yeah.
And this is partially why we have a collaboration with Chris Johnson and Ski, because they help us to visualize some of this stuff and sort some of this stuff out. We've been working with a graduate student named Ethan Kersner and a faculty member, Mariah Meyer, on some ways to start pulling this apart. But I'd sort of like to think of this more as neurally informed computing, as opposed to inspired computing.
When you look at this kind of complexity and you consider the question of that we've been talking about this morning, pictorial processing, higher level computing, what do you see? What kind of higher level computing do you see here?
Well, so vision has come and gone multiple times over 500 million years. You know, the first sort of fossil evidence of vision, trilobites, in the Cambrian, so 450 million years ago. And they developed very sophisticated compound eyes, and then, you know, vision has gone away, mammalians re-evolved it, plugged in rod pathways into cone pathways. So it's each visual system that has evolved has responded to a niche. And yet it's highly optimized over time, right? So our eyes, we've had, you know, 150 million years of evolution to sort of take, you know, the visual scene and process it with less than a couple watts of power. So it's highly energy efficient. It processes visual scenes and helps us sort visual information. And I think the important thing is once we understand what the network topology is, neural systems are good difference engines, right? So it may not matter what kind of information you put in. It could be visual information. It could be stock trading information. It could be internet packet routing information.
Comparing differences and pre-preparing the information for other types of treatment.
Yeah. And it could actually be an incredible cryptography one-time write-pad engine.
Sure, yeah.
So, and I've talked to some folks at Apple and Google about that. And, yeah, it's, there's a lot of potential for biologically informed computing.
Mhm.
Yeah.
And we should after, are there questions? We have people with questions? Here we go.
I don't know the numbers. I really appreciated the talk, but I think I'm about 100,000 of me with about 10 million of me here managing a conversation. And you just showed us how a bunch of those cells all talk together. The last slide that was up there, if you could put that back up again, that might be the picture of the network here at FiRe, not the one,
oh,
this one here.
This one.
This might actually be the FiRe community. I'm curious. And you're talking about in the me that's, you know, maybe 100,000 or 10 million of me standing here, my cells that are standing here. You're telling me how a certain layer is, a certain community is talking.
Yes.
Can you articulate how we best understand that conversation in that community?
So yeah, you know, part of the retinal neuroscience community for, again, 150 years has been trying to figure out how each individual neuronal type signals. So, you know, one of the things that we do is we plug electrodes, glass microelectrodes, into cells, and we listen to them. And you can stimulate them and sort of understand their response profiles to light, to darkness, to different pharmacologic conditions. And then other people look at network effects of that. So they'll plug other electrodes in downstream and stimulate the upstream neurons and try and understand how information flows through the retina. The problem is we don't have a good network topology to plug that information into yet. So we're kind of working in the dark. But you know, the fact that these networks are so complex tells us a couple of things. It tells us that there are a large number of potentially useful networks that we can use to process information out of. But then, you know, once we have the framework, then we can hang all the physiology and genetics and things like that into it. But, you know, so in a sense, understanding the edge node network topology is only the first step.
Maybe we have time
Yes,
for
sir.
one more.
Yeah, thanks. I appreciate you trying to tackle the complexity of the visual system. It's, you know, an incredibly complex system that you demonstrated. But I'm wondering if there's other people looking at, that's just one of our senses, right?
Yeah.
As humans. So if the idea of like looking at the theme of FiRe, like you were saying yesterday, is looking at big complex systems, like are there other people out there looking at different senses? So, you know, touch, smell, taste, and are you guys connecting? Could there be a connectome of all these different senses together, type of thing? So
Absolutely. So there's a bunch of different ways, I think, for augmented reality, for instance, that you can use alternative senses. Mark mentioned the tongue thing. That actually started as an Office of Naval Research project about 15 years ago for Navy SEALs to navigate complex three-dimensional environments without lights. Right? So they plugged them into sonars.
Yeah,
And it's a little grid of electrodes, and it feels like pop rocks.
yeah, right.
Remember pop rocks,
Yeah,
right?
Yeah.
But it projects a topographic map of the scene on your tongue, and so you can raster your head and pick things up and navigate. And so that's one potential sense that you can sort of augment if you understand the network flows. There's certainly hearing, there's vibration, there's balance, there's a number of different ways that we can sort of augment information flow. Absolutely.
Thank you. I think we have to wrap it up.
Thanks very much.
All right, really cool.
Thank you.
Thanks a lot.
All right, appreciate it.